Hi, I'm Albert Rivera, a graduate student at CSU, and in this presentation I will be going over my expertise on the technique that is known as stem cell culturing, while focusing on how this is tied into advanced microbiological research. This technique is used to maintain and grow stem cells in a controlled environment outside of the body, but automatically this technique gets a lot of bad reception from the public, so I want to talk about its advantages first. These cells are unique cells with the ability to differentiate into various cell types, making them valuable for research, regenerative medicine, and therapeutic applications. There are different types of stem cells, including embryonic stem cells and adult or somatic stem cells, with the embryonic being the first topic of debate and is under regulatory restrictions in some regions. But its advantages are what makes this such a valuable technique due to its variety of potential applications compared to other similar cell culture techniques. Some include the potential of stem cells to differentiate into a wide range of cell types, making them fundamental for regenerative medicine, like I said, valuable to drug discovery due to its being able to test against potential drugs, and even personalized medicine for individuals' genetic makeup. I can't glorify the advantages of this technique without going into its limitations outside of ethical concerns, two of them being the significant commitment of time and resources. Since it takes weeks to establish cultures and much attention to make sure they are adequately sustained, because if the culture is not properly controlled during differentiation, stem cells have a tendency of tumorigenicity, which is the formation of tumors. And along with this attention to detail, each colony needs to be dissected by hand for daily evaluation, which is depicted in the figures shown. This colony was sliced into about 100 pieces and cut into strips and then squares, as seen in figures A and B. A quick overview of this technique is starting with isolating the stem cell, then culturing it with a medium containing nutrients, adding a substrate or matrix that the cell could adhere to, or a layer of feeder cells, then, as depicted in the previous slide, passaging. This is where cells multiply and get split or, quote, passaged to prevent overgrowth and maintain their pluripotency, followed by differentiation for specific cell type applications, and of course quality control that helps ensure the maintenance of this precious culture, which afterwards could get cryopreserved at low temperatures for future use in a sterile environment. One way to optimize this technique is careful monitoring of the culture medium and adding sensors as the ones depicted in the figure shown, and then having computer programs analyzing what the extracellular and paracellular sensors pick up. In conclusion, the advantages of stem cell culturing vastly outweigh public deception, and in my humble opinion, this should matter more than anything else. And scientists are at work to perfect the use of using stem cells outside of embryonic stem cells. And here are my sources. Thank you. Have a good night.